The central dogma of molecular biology explains how genetic information flows inside the cells. It was proposed by Francis Crick in 1958. The process describes the transfer of information from the DNA to the RNA and then to the protein. This principle forms the foundation of molecular biology. Flow of genetic information. DNA stores the genetic code of an organism. RNA acts as an intermediate that carries this code. Proteins are the final functional products that perform structural and enzymatic roles. Thus, information moves in one-way direction, DNA to RNA and to protein. Step 1. Transcription, DNA to RNA. Transcription is the process of copying genetic information from DNA into RNA. It takes place in the nucleus of eukaryotic cells. Initiation. RNA polymerase binds to the promoter region of the DNA molecule. Elongation. The enzyme reads the DNA strand and builds a complementary DNA strand. Termination. When the end of the gene is reached, transcription stops. Processing. In eukaryotes specifically, the primary RNA transcript undergoes capping, splicing, and polyadenylation to become mature mRNA molecule. The mRNA then exits the nucleus and enters the cytoplasm for protein synthesis. Translation. RNA to protein. Translation converts the mRNA sequence into a specific amino acid chain. This occurs in the ribosomes. The ribosome binds to the mRNA at the start codon, which is AUG. Each tRNA brings an amino acid that matches the codon on the mRNA molecule. The ribosomes, they join amino acids together by peptide bonds. The growing chain forms a polypeptide, which folds into a functional protein. DNA replication, which is DNA to DNA molecule. Before a cell divides, it must copy its DNA molecule. This process is called replication. It ensures that each cell receives an identical copy of the genetic material. Now let's talk about beyond the central dogma. Initially, scientists believed that information flowed only from DNA to RNA to protein. However, later discoveries revealed exceptions. RNA replication, which is RNA to RNA. RNA viruses such as influenza and SARS-CoV-2 copy their RNA molecules directly using RNA-dependent RNA polymerases. Prions, protein to protein. Prions are infectious proteins that can cause other protein to misfold, leading to diseases like Creutzfeldt-Jakob disease. Importance of central dogma. The central dogma explains how genetic information is expressed and regulated. It helps scientists understand gene function, disease mechanisms, and drug design. Modern tools such as CRISPR use this knowledge to edit DNA and correct genetic errors. Thank you so much for watching the video. We will meet again in the next one with a new and engaging topic. Thank you so much for your support.